Why is there no MIS vaccine for EBV? Many people wonder why, despite knowing that the Epstein-Barr virus is strongly linked to multiple sclerosis, there is no vaccine to prevent it. The connection between EBV and MIS is well established through scientific studies showing that infection with EBV increases the risk of developing MIS by more than 30 times. Yet, creating a vaccine to stop MIS from ever happening remains a complex challenge. One major reason is how tricky EBV is to target. Once someone gets infected, usually during childhood or teenage years, the virus hides inside B cells, a type of immune cell. It can stay dormant for life, making it hard for vaccines to completely eliminate or prevent the virus from reactivating. The immune system has a delicate balance when fighting EBV because it needs to control the virus without attacking the body's own tissues. This is especially important because MIS involves the immune system mistakenly attacking the protective covering of nerves, called myelin. Scientists believe that EBV triggers this autoimmune response through a process called molecular mimicry, where parts of the virus resemble nerve proteins. Designing a vaccine that prevents infection or reactivation without causing harmful immune reactions is a major scientific hurdle. Another complication is how common EBV is. Nearly 95% of adults worldwide carry the virus, often without any symptoms or health problems. Most people infected with EBV never develop MIS. The disease usually appears years after the initial infection, which makes it difficult to prove exactly how EBV causes MIS or to determine the best time to vaccinate. To develop an effective vaccine, researchers need to figure out how to prevent the initial infection or alter the immune response early enough to reduce the risk of MIS. This requires long-term studies to see if vaccination can truly make a difference. Currently, vaccines aimed at preventing EBV are still in the experimental stage. Researchers are working on vaccines that could stop infectious mononucleosis, a common illness caused by EBV, and possibly reduce other EBV-related diseases, including MIS. If these vaccines prove safe and effective, they could potentially prevent many cases of MIS by stopping the virus before it triggers the immune system to attack nerves. But it will take time to complete clinical trials, assess safety, and understand how well these vaccines work in the real world. Further research is also needed to fully understand how EBV influences the immune system to cause MIS. Scientists are studying the detailed mechanisms behind this process, including how the immune response changes after infection. It is crucial to ensure that any vaccine developed does not unintentionally worsen autoimmune responses or cause side effects. In summary, the reason there is no vaccine for MIS targeting EBV yet is because of the complex nature of the virus and how it infects and persists in the body. Developing a vaccine against a virus that establishes lifelong latency is a significant scientific challenge. The high prevalence of EBV in the population means that most people carry the virus without developing MIS, making it harder to identify who would benefit most from vaccination. Ongoing research is promising, and future vaccines or antiviral therapies may help prevent MIS by targeting its viral trigger. Until then, MIS treatment continues to focus on managing symptoms and supporting immune health under the guidance of healthcare professionals. Always consult with medical experts for personalized advice and stay informed through trusted health sources.